Anaerobes of clinical significance. Anaerobes are able to replicate in the absence of oxygen. Obligate or strict anaerobes are killed in the presence of oxygen almost immediately. Capnophilic require an increased carbon dioxide and slightly decreased oxygen level in their environment in order to grow well. The conditions in a capnophilic environment are still sufficient to allow aerobic organisms to replicate. Microaerophilic oxygen is reduced to 5% or less. They still need oxygen for certain life processes, but not as much oxygen as like an obligate aerobe would require. Facultative anaerobes preferentially use oxygen as an electron acceptor if it's available, but they can grow in the absence of oxygen, just slower. Aerotolerant organisms can survive in short exposure to oxygen but are not able to perform metabolic processes unless they're placed back into an anaerobic environment. So this is a table that you can pause the video and review. Molecular oxygen has a direct toxic effect to some anaerobes. Anaerobes lack enzymes to break down those toxic products. Main toxic products are superoxide anion, hydrogen peroxide, and hydroxyl radical. They require antioxidants in order to break them down, but they don't have those antioxidant substances and they don't have appropriate enzymes to break those down. Strict aerobic and facultative anaerobic bacteria can protect themselves. They do possess enzymes to break down those toxic products. Superoxide dismutase can break down those superoxide radicals, and catalase can break down hydrogen peroxide. When it does break these down, it breaks it down into non-harmful substances. And you can see at the end of this reaction, you're left with water and oxygen. Anaerobes can be exogenous or endogenous. Exogenous organisms exist outside of the bodies of animals. Infections develop when the agent is ingested or has gained entrance through trauma. Exogenous infections are usually caused by Clostridium species. Clostridium species are gram-positive and spore-forming. Endogenous organisms exist inside of the bodies of animals and become sources of infection. Examples include Bacteroides, Porphyromonas, Prevotella, Clostridium, Propionibacterium, and Anaerobic cocci. Propionibacterium is no longer the commonly accepted term for it. They've changed the name again and updated it to Cutibacterium, and I'll reference that later on in the slides. The qualifying exam may use the old or the new name, or they may put the new name first and then put Propionibacterium in parentheses. So here's a table of endogenous anaerobes commonly involved in human infections. You can pause the video and read through this list. And here's the incidence of anaerobes at a university medical center. So this just kind of shows you the different types of anaerobes and the ones that are most commonly encountered. And you can see Bacteroides fragilis is the highest and most commonly encountered. At specific anatomic sites, in the skin, you would see Cutibacterium acnes, that's formerly known as Propionibacterium acnes, Peptostreptococcus. In the respiratory tract, Prevotella, Porphyromonas, Fusobacterium, and Anaerobic cocci. In the GI tract, Bacteroides, Bifidobacterium, Clostridium, Eubacterium, and Anaerobic cocci. And the genitourinary tract, Fusobacterium, Prevotella, Bacteroides, and Lactobacillus. Anaerobes produce a variety of virulence factors. Trauma to protective barriers allow anaerobes to enter in through the skin and mucous membranes. Vascular stasis. So if there's vascular stasis or a decrease in blood flow to a particular area, then there's not going to be a lot of oxygen in that area. That creates a slightly anoxic environment, which is conducive to growth and multiplication of any anaerobe that might be present at that site. Similar results can occur in instance of tissue necrosis. So anaerobes are especially bad because they don't like oxygen. So what they do is if they get into an infection, they try to kill off all of the neighboring cells because if the cells are dead, they're not going to be bringing oxygen into the body. Their ultimate goal is to completely kill the whole organism so that they can eat you. Obviously, we have medical intervention that hopefully helps slow that down and prevent that from happening. But that's really the goal of anaerobes. They want to create an oxygen-free environment around them. And they want to do so by killing off and causing widespread tissue damage so that oxygen doesn't flow through those tissues. Indications of involvement of anaerobes in infectious processes. Infection in close proximity to a mucosal surface. Presence of foul odor. So that's one of the keystones of an anaerobic infection, is the odor coming from the actual patient is a really putrid and foul smell. So you can kind of smell the wound or smell the area, and it's a horrible smell, and you can kind of assume that it's anaerobic involvement. Presence of a large quantity of gas, black color or brick red fluorescence, or presence of sulfur granules. So now we're going to go through commonly encountered anaerobes, and we're going to start with gram-positive spore-forming anaerobic bacteria or bacilli. So in this category, predominantly Clostridia, Clostridium, or Clostridioides. All species are capable of producing spores. Some do so readily, some require extremely harsh conditions in order to produce spores. Spores are not observed in gram-stained smears of clinical specimens containing Clostridia or in smears of colonies from an agar plate unless the culture has been incubated for many days. The species is usually differentiated based on the location of where the spores are. So terminal spores, C. tetani, so you'd see the spore at the kind of end of the bacterium, while C. sordellii has it in subterminal, so just before the end. You could see there's bacterial cell, spore, a little bit more bacterial cell, while in this instance over here, they're all the way at the tip. Clinical infections most frequently encountered in exogenous anaerobic infections or intoxications. Modes of transmission, ingestion, or open wounds that have become contaminated with soil. Almost any Clostridium species can cause wound or abscess infection, usually part of mixed biota, and most can be isolated from the blood in cases of bacteremia. One Clostridial infection of endogenous origin, antibiotic-associated pseudomembranosus colitis caused by C. difficile. C. difficile was one of those kind of like Cutibacterium and Propionibacterium, used to be called Clostridioides or Clostridium difficile, and now it's Clostridioides difficile. Clostridium perfringens food poisoning. So there's type A, when you ingest the enterotoxin causing diarrhea and cramps. It's relatively mild and usually self-limiting. Type C is enteritis necroticans, and that's acute onset of severe abdominal pain and diarrhea, which is often bloody and may be accompanied by vomiting, followed by necrotic inflammation of the small intestine, at times leading to bowel perforation. Without treatment, it can be fatal. Clostridium perfringens food poisoning is pretty common. In certain parts of the world, it's more common than like what we would think of as E. coli food poisoning. Botulism. There's foodborne, where you have ingestion of preformed botulinum toxin produced in the food by C. botulinum. There are 7 antigenetically distinct botulinum toxins, A to G. Only types A, B, and E are associated with human disease. The botulinum toxin is an extremely potent neurotoxin. Only a small amount is needed to produce paralysis and death. But it's also used as a drug to treat watery eye chronic migraines, and it's a beauty enhancer. Botulism are the Botox injections. Sources of infection, home canning, and home cured meats. So what happens is if you don't properly decontaminate the food as you're preparing it, you put it in a can and then you seal it, and you're sealing it in an anaerobic environment. So if the bacteria was trapped inside and it's trapped in an anaerobic environment, it's going to grow and multiply, and it's going to produce a whole bunch of that toxin. Then you open up the can, you cook the food inside of the can, or maybe you don't, maybe you eat it right out of the can, who knows. Um, but there would have been some gas formed, so the can would have looked a little weird. It would have been stretched a bit from the pressure of the gas that was produced. But if you ignore that, open it up, and go dig in there, you'd be eating preformed toxin, and that can be deadly. Symptoms 2 hours to 8 days later. Infant botulism, ingestion of spores and contaminated honey. This is why they say you should never feed your child honey unless or until they reach a year of age or older. Colonize the colon and produce toxins. Wound botulism, contamination of wounds with spores of C. botulinum. Similar clinical manifestations to those in the foodborne origin. Botulism toxin and C. botulinum are considered as potential bioterrorism agents. Laboratories are required to report suspected cases to public health laboratories. Tetanus. The causative agent is Clostridium tetani, attributed to tetanospasmin neurotoxin production. Inhibits neurons by inhibiting neurotransmitters. It results in a spastic type of paralysis, with continuous muscle spasms leading to trismus or lockjaw. Rhizus sardonicus, distorted grin, and difficulty breathing. It can cause the muscles to contract so much that you can actually break your jaw. It will cause your back to arch and you can break your back. It can cause constriction of the muscles that are involved in breathing and prevent you from being able to breathe. Spores in the environment enter the skin through puncture wounds. Symptoms typically appear approximately 7 days after injury. An incubation period can range from 3 to 21 days. Length of incubation is related to the distance from the injury to the central nervous system. Muscular rigidity, usually in the jaw, neck, and lumbar region. Difficulty swallowing results from the muscle spasms in the pharyngeal area. Rigidity of the abdomen, chest, back, and limbs. Tetanus is no longer a common disease in the U.S. because we use the vaccine to prevent against it. Therapy requires injection of antitoxin, muscle relaxants, and intensive therapy. Myonecrosis. This is also known as gas gangrene. Contaminated wounds from trauma or surgery caused by several species of Clostridium. So perfringens, histolyticum, septicum, novae, and bifurmentans. Grow, multiply, and release potent exotoxins. Alpha toxin causes tissue necrosis and deeper invasion, causes death of the tissue that can require amputation. Symptoms would be pain, swelling, fluid-filled blisters, serous discharge or discoloration, and tissue necrosis. The onset and spread of myonecrosis can be rapid and extensive. Surgical debridement of the necrotic tissue is often required. If treatment is delayed, amputation is not uncommon. Bacteremia, bacteria in the blood. C. perfringens is the most common. C. septicum is a marker organism for malignancy of the GI tract. C. tertium and C. bifurmentans, both isolated from patients with underlying disease. Antibiotic-associated diarrhea caused by C. difficile, Clostridioides difficile. It's the most common isolate in antibiotic-associated diarrhea. Basically, patient comes in, goes on an antibiotic for a period of time. That antibiotic wipes out their good bacteria, allowing this bad guy to overgrow. It can cause pseudomembranosis colitis, necrosis of the colonic mucosa, and bloody diarrhea. It develops when normal flora are destroyed by antimicrobials that do not affect Clostridium. Increased growth causing toxin production. So there's an A and B toxin that can be detected by immunological and molecular tests. Commonly transmitted as a nosocomial infection. It's transferred among patients, hands of hospital personnel, and frequently is increasing in outpatients who have received antimicrobial therapy. Because it's a spore former, the spores can persist in hospital environments, and it's really difficult to get rid of. This is actually what my grandma died of. So my grandma had pretty significant stroke following a heart procedure, and she was in a long-term healthcare facility for a period of time. And in that long-term healthcare facility, she ended up getting C. diff. And C. diff caused so much nutrient loss that eventually she wasted away and died from this. There was nothing that they could give her to decrease the extent of the C. diff that she had, and she was suffering a great deal from this in the end. So C. diff is definitely something that we need to do a lot more research on, and we need to look into possible alternative cures to help prevent the spread of C. diff in hospital clinics. Now, we do have these like big UV machines, and they look like little UV robots that kind of go from room to room and spread their UV rays around. Well, endospores are resistant to UV. So the UV radiation will kill vegetative bacteria. It doesn't really do much for the spores or the endospores that are left behind. So need to figure out different ways to control this, because someone goes into a hospital when they're sick, so they're already slightly immunocompromised. Now we give them C. diff, it's all over the hospitals. Many people are naturally carriers, and they say like 90% of healthcare workers are already carriers for C. diff. So it's pretty much everywhere. It takes consumption of an antibiotic in order to reduce the natural flora, or it takes some major medical event to reduce the natural flora, and that allows this thing to overgrow. Gram-positive non-spore forming anaerobic bacilli. So these ones don't form spores. Actinomycetes, Bifidobacterium, Agarthella, Mobiluncus, Propianobacterium, and Lactobacillus. Actinomycetes, chronic granulomatous infectious disease characterized by the development of sinus tracts and fistulae which erupt to the surface and drain pus that may contain sulfur granules. Diagnostic procedures, gram stain preparations of pus draining from sinuses that reveal non-spore forming thin gram-positive bacilli that frequently exhibit branching. So this one looks like something's coming out of their face. So you see these big bumps that are eroding from the sinuses onto the surface. This is what it looks like under a microscope. For the longest time, they even thought that this was a form of like nocardia, or it was branching like fungal elements. Many cases of actinomycosis occur in maxillary region with draining in the neck and thorax. Cases can also be seen in the female genital tract. Most common cause is Actinomycetes israelii. Other possible causative agents, Propianobacterium and Bifidobacterium. Bacterial vaginosis is thought to arise from a change or shift in the endogenous microbiota of the vagina, result in decreased normal lactobacilli, and an overgrowth of other endogenous anaerobes of the vagina. Clinical features: gray-white, homogeneous, malodorous vaginal discharge with little or no discomfort or inflammation. Associated with increased risk of acquiring HIV infection, experiencing adverse outcome in pregnancy, possibly associated with pathogenesis of pelvic inflammatory disease. Lactobacillus, gram-positive, highly pleomorphic bacilli. There's more than 100 identified species, aerotolerant anaerobes, widely distributed in nature, in foods, and is part of normal biota in the human mouth, GI tract, female genital tract. Play an important role in the health of the female genital tract. Lactobacillus acidophilus complex constitutes most of the lactobacilli of a healthy vagina. Specimens of choice, urine, genital cultures, systemic infections are rare, but when present, typically originate from the oral cavity. Serious infections in immunocompromised patients can be bacteremia and endocarditis. Anaerobic gram-negative bacilli. So now we're on to the gram-negatives. We've got Bacteroides fragilis, which is the most commonly encountered anaerobe in the clinical setting. Prevotella, Porphyromonas, and Fusobacterium. All part of microbiota of the oral cavity and GI and GU tracts. As I mentioned, B. fragilis is the most common isolate. It's found predominantly in blood, ulcers, intra-abdominal abscesses, and others. Brain abscesses would be Prevotella, Porphyromonas, and Fusobacterium, all endogenous microbiota of the oral cavity. Anaerobic cocci. Group members: Peptostreptococcus, Anaerococcus, Finagoliad, or Goldia, uh, Gallicola, Parvimonas, and Peptonophilus. Only implicated pathogen is Veillonella. Anaerobic cocci are weakly pathogenic. They may be found in a wide variety of infections. Examples include brain abscess, aspiration pneumonia, lung abscess, gingivitis, other periodontal disease. Periodontal disease is huge with anaerobes, so lots of anaerobic bacteria like to live inside of the teeth because it's a pretty oxygen-free environment. Finagoldia magna is the most pathogenic anaerobic cocci. It's associated with endocarditis, meningitis, and pneumonia. Peptostreptococcus anaerobius is linked to polymicrobial abscesses of the brain, ear, jaw, pleural cavity, and other sites. Evaluating the significance of anaerobic gram-positive cocci isolated in clinical specimens is often difficult. Specimen collection or selection, collection, transport, and processing. When physicians suspect an infection involving anaerobes, so let's say it looks black in color or it has a really bad smell, the specimen collected must be from the actual site of the infection and not just a swab of the mucosal surface. So you actually have to get the swab underneath the surface of the skin, deep inside of where the infection is originating from. It's important that there's no prolonged exposure to oxygen, and it must be transported as quickly as possible to the laboratory under anaerobic conditions. These are acceptable specimens for anaerobic bacteriology. Your qualifying exam is going to say, throat swab was submitted for anaerobic culture. How should you process this specimen? Well, if we go here, throat swabs are unacceptable specimens for anaerobic bacteriology. So one of the options would be like, would you streak it to SBA, streak it to chocolate agar, gram stain it, and then streak it to different plate media, or reject the specimen as not appropriate. So you would have to be able to figure out that, well, if it's in an area or a surface of the body that commonly encounters oxygen, it would not be an appropriate specimen for anaerobic culture. So go ahead and pause the video and review these two slides to see which are acceptable specimens and unacceptable. Regardless of the type of specimen submitted for anaerobic bacteriology, it must be transported and processed as rapidly as possible with minimum exposure to oxygen. Specimens are usually collected from a warm, moist environment that is low in oxygen. It's important to avoid shocking the anaerobes by exposing them to oxygen or permitting them to dry out. Specimens should not be refrigerated, and the amount of time they remain at room temperature should be minimized. Needle and syringe aspirates. Less contamination and can easily expel any air that's in them. Best to inject the specimen into pre-reduced anaerobically sterilized media, PRAS. Once in the laboratory, you vortex to mix it and plate it to the appropriate isolation media. Use a few drops of on-PRAS media and streak for isolated colonies. Vital since most anaerobic infections are polymicrobial. Need to isolate colonies to verify relevant organisms. So this is what you would put swabs into. So you would swab and then you would put it into one of these transport systems. The transport system would reduce oxygen. If you get just a normal swab sent out to the lab, typically we'd have to reject that if it's for anaerobic culture. Anaerobic culture also has to be specially ordered by a physician. We don't normally just do anaerobic culture. One that we would incubate is the blood culture tubes. We always do one aerobic and one anaerobic blood culture tube. But unless it turns positive for anaerobes, we typically wouldn't go and work it up. If both of those blood culture tubes turn positive, we would only work up the aerobic culture. We typically wouldn't work up both of them, as long as we think that the same organism is growing in both of those. It's highly unlikely that there would be more than one type of bacteria growing inside of the blood. It's possible though, so if we do happen to look at the blood culture tube under the microscope and we see different types of bacteria, like let's say the aerobic one is gram-positive cocci and the anaerobic one is a gram-negative rod, then we would go ahead and work up both of them, because then at that point, obviously, they have more than one thing growing in their blood. Swabs. As I mentioned, not appropriate for anaerobic culture, should only be used if aspiration is not possible and biopsy specimen is not available. When swabs are deemed necessary, they should always be transported under anaerobic conditions. Once in the laboratory, they should be placed in a tube containing about 5% or 5 mils of sterile thioglycolate broth. Swab should be vortexed vigorously to remove material from the swab and then pressed firmly on the inner wall of the tube. Remove as much liquid as possible. Media are inoculated to the media using liquid suspension. Alternatively, a swab transport system can be used. Small pieces of tissue can be placed in an anaerobic transport tube vial containing PRAS medium. Larger samples can be sent in a sterile container with sterile wet gauze. If there's a delay in transport expected, tissues may be transported in pouches containing oxygen-free atmosphere. To process a piece of tissue or bone fragment, 1 ml of sterile thioglycolate broth is added to the sterile tissue grinder. Piece of tissue or bone fragment is homogenized until a thick suspension is obtained. Ideally, this process is performed in an anaerobic chamber. If not available, grinding must be done as quickly as possible in a biological safety cabinet. The suspension is used to inoculate media. Blood. Aseptic specimen collection and subsequent inoculation of both anaerobic and aerobic culture bottles. Vein puncture site is prepared for specimen collection with a bactericidal agent combination with 70% isopropyl alcohol. Once inoculated, bottles should be rapidly transported to the laboratory where they are incubated. Anaerobic chambers provide oxygen-free environment to work with anaerobic cultures. Remember, oxygen kills strict or obligate anaerobes, so we can't just open them up on a bench top and start looking at them if we expect to have to subculture them or do further tests where we need them to be alive for those tests. Required safety measures: always wear gloves. Biosafety hoods should be used when manipulating samples. You do macroscopic examination of the specimen, preparation of gram stain smears for microscopic examination, inoculation of appropriate plated and tube media, and anaerobic incubation of the inoculated media. Choice of media: CDC blood agar is the best recovery. It contains sheep red blood cells, vitamin K, hemin, and yeast extract. And here's a table of different types of media that would be used for primary setup. And this is what it looks like. Specimens submitted for anaerobes typically also have a request for aerobic culture. And if it has an aerobic culture request, you would put those on chocolate beep, AP, blood agar plate, McConkey, and in some laboratories, CNA, selective media for gram-positive bacteria. Here's the culture results from a hypothetical wound specimen. It's kind of interesting to take a look at, so you can go ahead and review this diagram. So alternative inoculation in an area with a suitable nitrogen gas holding system, a jar, box, or other small chamber. It is important not to keep inoculated plates out at room temperature more than an hour. Maintaining environment keeps cultures viable. The gases need to be predominantly hydrogen, CO2, and nitrogen. Catalysts: palladium-coated alumina pellets remove any residual oxygen. So this is what some labs use. So it's this chamber where you would seal it. You would put one of these gas packs in there, and the gas converts and removes all of the oxygen from the environment. This is what another system looks like. So this is actually attached to a gas regulating system. And then this is a specialized hood that would be used to evaluate and work up anaerobic culture plates. So typically when we look at our anaerobes, so we will plate them in standard conditions, and after they've grown in those standard conditions, we would want to look at them and see if we have to do any subculturing or any further workup from there. And if we do, we would want to open those up in an anaerobic environment. So what you do is there's a door here. So you would open up the door and you would put the items inside of the door. Then you would press the button and it would exchange out the environment, removing the oxygen. So here or here is where it would enter. So when the outer door is sealed, now you would put your hands into the gloves and open up the inner door, and you would bring everything into that anaerobic environment inside. Then you can open up the jar, expose it to the atmosphere inside of here because it's an oxygen-free atmosphere. And here's an anaerobic bag. So an example of a bag that would keep the anaerobic environment. So indications of the presence of anaerobes, we already mentioned the foul odor, characteristic colonial morphology on anaerobic agar plates, but not on aerobic culture plates. Unique morphology on gram-stained smears of clinical specimens or isolates, good growth on Bacteroides bile-esculin BBE agar, double zone of hemolysis on blood agar, brick red fluorescence on kanamycin vancomycin laked blood agar. Processing colonies, describe the colony morphology, the gram stain, set up the aerotolerance test, and inoculate pure culture or subculture plate and add appropriate discs. So here's a diagram for the initial identification of anaerobic isolates based on the gram stain. Fluorescence. So under UV light, Porphyromonas and Prevotella look brick red. So you can see this. And you'll see on the qualifying exam, it'll say which one is brick red fluorescence, and it'll give some potentials. I've seen Prevotella on there more than I've seen Porphyromonas. And then the chartreuse one would be Fusobacterium and C. diff. Typically, we don't culture for C. diff. C. diff is identified based on the presence of toxins. So you can see the list and the color of the fluorescence. And take some time to pause the video and review the list. Other tests include the catalase, spot indole, rapid urease, rapid motility test. Antimicrobial discs, vancomycin, kanamycin, and colistin discs are used. Occasionally we'll use SPS discs, bile discs. And here you can see the breakdown of what the different discs are used. The egg yolk agar lecithinase, C. perfringens would be positive for growing on this, so opaque zone around the colony. Lipase, Fusobacterium would be positive for this one. So here you can see a positive lecithinase and a positive lipase. So B. fragilis on anaerobic KVLB and BBE agar, and B. wadsworthia on BBE agar. So if you look at it and you can see this black halo around the growth, then you know it's likely to be Bacillus fragilis. Fusobacterium nucleatum looks like this. So elongated, fusiform, like filamentous looking like rods. So this is pretty characteristic, and this is something that you may encounter in the future. So make note of the gram stain result of Fusobacterium. Biochemical based multi-test systems, there is an API system. You inoculate different wells and you would incubate that anaerobically and then do it biochemically. There's also enzyme-based systems, and then there's the MALDI mass spectrometry system that can identify them. This is a picture of the rapid ANA2 preformed enzyme system for the identification of anaerobes. And then 16S ribosomal RNA sequencing. So if we can't identify it by other means, we would have to go to the molecular diagnostics route. And there's going to be a lot of tables that I'm going to just kind of go through real quick. You can definitely take some time, pause the video, and look through them. And these are just diagnostic schemes for being able to identify the different species. So the non-spore-forming gram-positive bacilli, actinomycetes, Bifidobacterium, Lactobacillus, Proprianum, and Cutibacterium, molar tooth colonies of A. actinomycetes israelii. Bifidobacterium, variable in shape, ranges from coccobacilli to long branching rods. Ends may be pointed, bent, club-shaped, spatulated, bifurcated, or forked. Cells may appear in singly or in chains, V arrangements, palisades, clusters. The colony may come in different variations as well. So this is Cutibacterium acnes. And another one of those phenotypic characteristics of various anaerobic gram-negative bacilli. And a diagnostic scheme. Antimicrobial susceptibility testing. It's performed when anaerobes are recovered in brain abscesses, endocarditis, infection of a prosthetic device or vascular graft, joint infections, osteomyelitis, or bacteremia. Highly virulent and or commonly resistant organisms are listed here. Primary choices and supplementary choices of antibiotics are listed. But there's issues with anaerobic testing of isolates. So lack of reproducibility, the failure of some anaerobes to grow appropriately. So some anaerobes, because they're anaerobes, they don't grow well in these systems where we need to evaluate for the presence of antibiotic susceptibility. If there's no growth in anything, then we can't just assume that it kills the antibiotics are super successful. We have to actually get the thing to grow before we can determine antibiotic susceptibility. Lack of comparability between the methods, cost, and complexity of testing. So the agar dilution method is the gold standard, but it's labor intensive. It's not practical for the use in smaller clinical laboratories and primarily used in reference or large laboratories. Broth microdilution only for members of the B. fragilis group.